Hi, everybody. I'm Jack Van Horn. This
This
is
is
exciting for me to come. This is my very first Neurohack Academy, and yet I'm from the great state of Washington. Seattle is my hometown. I grew up on a little island you have to take a ferry boat to. For those of you who are familiar with Puget Sound region, Vashon Island is my home. I just spent a week here with some family and took part in the Vashon Island Strawberry Festival, which is sort of like the community coming together.
It's like a big
high school reunion. And it was a lot of fun. And so I love coming back to Seattle. And it's really exciting to take part in this event. One I have been really excited to take part in. I'm really glad that I was offered the opportunity to do that. I'm Jack. I'm at the University of Virginia, as Ariel said.
Sorry.
I've been a number of different things in my career, and currently I am jointly appointed in the Department of Psychology at the, yes?
Can I like with your address? I see a lot with raised hands. I just want to check.
Uh-oh.
Yeah, I'm sorry. We just like online participants. We can hear really quite strong echo.
Oops. Is this microphone
Yeah.
on?
Yeah, I
I
think
think
like
that's
two
for everybody
microphones.
online.
Yeah, probably two microphones in the classroom actually is on, so maybe.
Or
Just mute
probably
one.
because you are
Everyone,
unmuted.
is that better?
Yeah,
Online
that's
people?
better.
Yeah, now it's
Okay,
good. Thank you so
apologies.
much.
Thanks.
There we go. Okay, apologies about that, everybody. So again, I'm Jack Van Horn from the University of Virginia. I'm jointly appointed in the Department of Psychology, as well as in our new School of Data Science. The new School of Data Science just opened a new building just only a couple of months ago. So and we're actually the newest school at the University of Virginia, which is pretty exciting. And it means that we're kind of making stuff up as we go, which is also kind of exciting. But over the years, I have had an opportunity to be part of many different kind of early initiatives and teaching courses in neuroimaging and neuroscience and kind of computational methods at Dartmouth College, at UCLA, at USC, and now at UVA. So education is something I take very, very seriously and do a lot of work in. And so I have culled a number of different things from some of the undergraduate neuroimaging courses that I have taught, the graduate courses, as well as like fundamental research related stuff, to kind of cobble this together to kind of talk to you about things that I think are important as we're thinking about the brain, as we're thinking about the science of data, computational methods, machine learning, all those things, and how we put it all together. And so what I want to talk about today is break this kind of talk down into a number of different, oops, let me move this thingy if I can. Move you. Can I shrink him all together? Was it hide that? Go click on that, turn up volume thing. Just try to get everything out of the way so everybody can see. Okay, to kind of break down my talk into kind of the brain science is a data science. I firmly believe this. I'm going to try and give you an impassioned argument about why that's the case. Talk a little bit about a brief history of neuroimaging databases, what might seem to you like something that everybody has just always done. The truth is more stranger than fiction on that front. I thought it would be interesting for you to hear that story. Talk a little bit about from data to models and then back again, and why I think that's kind of important and why we're at this stage right now, and then to talk about simulating systems as another way to move computational methodologies forward and do this kind of via a digital twins kind of an idea, and then reach some conclusions for you. I hope you'll get a number of take-home messages that hopefully will guide you as you're thinking about these projects you're going to be undertaking, some of the things that you're going to be bolting into those, and how you might make them that much more compelling and innovative. So I firmly believe that the brain science is a data science. This is kind of I don't know, something I've just arrived at over the years and thinking very deeply about this, really because the source of all data science, the source of all big data is this thing right here, this thing that we all have between our ears and that we are in the act of studying right now, Without the brain, there would be no big data. There would be no data science. There would be no computational methods, right? Human brains have thought this up, and we've organized ourselves in such a way to actually figure out how to encode data, how to analyze it, how to make sense of it. So this shouldn't be too surprising. But let's think about this for a little bit. Why is the brain kind of special? Well, this is a very famous graph about the encephalization of the brain. Whoops, gotta get rid of that guy. Encephalation of the brain, which is the expected brain size. So this is, again, very famous image, comes from Harry Jarrison from back in the 1970s, where he plotted log body weight in kilograms against log brain weight in grams. And you get this, what's called an allometric relationship, where you have this kind of little equation here is describing the slope of this line here, again, on log scale. But one of the things you see here is that the human brain in modern humans is actually deviates quite substantially from this expected brain weight to body rate relationship. In contrast to other mammals and other species of animals, even though they all share the same kind of 2/3 slope coefficient here, they're all kind of angled at the same rate. The humans are well above where you would expect to see them via their body weight. It's kind of really quite an incredible graph. And when you look at things like, how human evolution has happened over the years, it's really only in the last 2.5 million years that the size of the human brain has really exploded in terms of its size. When one looks at something like pan troglodytes here, that's 400 cubic centimeters worth of brain. In contrast to modern Homo sapiens, which is about 1,300 cubic centimeters. This is actually quite a sizable increase. And it's not just that it just gradually increased. There was a jump. There was a major leap in the size of the brain over human evolution. Now, many people have posited about why this might be. There might be some genetic influence. Very likely. There might be different things about how groups of individuals came together to create little social structures. So-called gene culture coevolution is one possible model. Changes in the diet, for example, gave more energy to a very energy-hungry organ to produce and make it even larger, among a number of other technological factors. But one thing that people don't often talk about is the brain's ability to calculate. And throughout human history, there have been a number of pieces of evidence which have emerged, which indicate that the human brain is really quite special in terms of being able to calculate things. Things called the Ishango bone, for example, which is a Paleolithic era artifact, which was found in the 1950s in Uganda, indicates that there are little markings on these little bones here that indicate an attempt to count something and then calculate against it. This is the earliest known calculator that apparently is in the human record. A number of centuries later, things like the abacus kind of emerged out of Asia to be able to calculate, to do math. This was very important when one is doing commerce, being able to keep track of how much things cost and how much you need to kind of keep track of that sort of thing. Being able to come up with accumulations of knowledge, such as in the Library of Alexandria, were an enormous resource for scholars and clerics and others to be able to have a central location where all knowledge existed. And even things like the Antikythera mechanism have been posited as being an early attempt to create a computer. It's something that would actually resemble something that we might think of as a mechanical computer. All these things are really kind of interesting because if you think about the level of human intellectual achievement, all of these things involve some ability to take information and to calculate against it. Whether you're part of a symphony orchestra and you're counting out the measures of your symphonic thing that you're doing, whether you're looking at genomics, the whole field of bioinformatics emerged out of doing computation against genetic information. Whether or not you're dealing with Maxwell's equations, which we'll come back to, Newtonian physics, whether you're dealing with high ideals of government and how things are meant to be, of, by, and for the people, involve some idea of counting. and making measurements and computing against it. Whether it's flight, we've only been able to fly for a little more than 100 years. But in that time, we've not only have we taken off from the sand dunes of North Carolina, we've landed people on other celestial bodies. This is an enormous idea, right? To be able to take not only a single individual or single individuals and figure out how to operate a lighter than air aircraft to within 100 years, less than 100 years, be able to propel somebody collectively with a collective group of brains to another celestial body. It's really quite incredible. Thinking of high energy and celestial and astronomical physics, doing art, architecture, everything are all sort of stuff that's like human brains came up with. It's really quite remarkable when you think about it. And scientific data, we have come up with all manner of ways to collect data, to measure it, to collect it not only in an MRI scanner, which we're all probably a little familiar with, but to be able to point telescopes at astronomical phenomenon light years away and be able to gather that data. It blows my mind that we're able to go and do that, and that data is transmitted down to our planet, and we're able to analyze it in a matter of, minutes. It's really quite incredible. Being able to look at weather systems, think about it, 100, 200 years ago, storms happened, right? And you didn't know it was coming. Now we have plenty of early warning systems that allow us to prepare and probably save lots of lives. This is an enormous amount of how humans are using large-scale data to do things which are useful. So really, my question is the brain to blame for data science? And I think that it is. I think that we have arrived at a time where it's easy to collect lots of data in a short amount of time. In brain science, we do not collect less data. We continue to collect more data at ever finer spatial and temporal scales of resolution. And I think that if you don't have the brain, you don't have data science. Now, what's really interesting is, as I mentioned, we don't collect less data in the neurosciences. We continue to collect more data. And here's just some data which kind of exemplifies this for me. When I started in human neuroimaging, I was asked by my lab director to go and buy the hard disk to store all of our vast sums of medical imaging, brain imaging data. And so I went out and I went shopping. I was not much older than some of you. I went out and went shopping, and I bought the largest freestanding hard disk that you could buy. It was 4 gigabytes. And I thought, this is infinity. I will never fill this up. This is massive. And people used to come by the lab and they just to gaze upon this beast of computer storage. This image that I'm showing you now is collected on a CT scanner. It's actually much larger than that hard disk that I bought back in 1990, whenever. And it allows you to, this particular data set allows you to continue to zoom in and zoom in and zoom in all the way from the whole brain, all the way down, as you'll see, to kind of white matter fiber pathways. And very soon we'll be able to zoom in here even more. You're almost seeing individual cell bodies using CT imaging. This is in a post-mortem brain, but one thing which is kind of interesting about neuroimaging is never say never. I remember when I first started in the field, I'm doing fMRI. Many of you are probably, if you're fMRI people, you're familiar with the parameter, the TR, right, or the repetition time, basically the sampling interval, temporal sampling interval. And I had a very senior colleague say, "Oh yeah, I don't really see us ever having a TR less than 4 seconds." That just blows my mind now. We're able to get sub-second resolution. And so if you just wait long enough, somebody's going to come along and they're going to show you how to get more data per unit time and per unit space. Of course, any single modality isn't necessary, nor is it sufficient to be able to explain and characterize something as complicated as the brain. Fortunately, in medical imaging, we have a lot of different technologies we can bring to bear to understand not only the spatial, but the temporal, but the neurochemical, the spectral, and a variety of other different methodologies. So all of these different methods up here are available on a single instrument. So you can use MR to get all of them. Of course, if you're also including things like PET imaging or various other like CT data, for example, you have other modalities as well. And when you look at something like traumatic brain injury, this is from a paper I did a few years ago, looking at how all these different modalities are super important for being able to look at something as complex as traumatic brain injury. Each different modality tells a different story about that data. No, there's no single smoking gun. You have to look at them all if you really want to get a complete picture. And this includes not only MRI, but PET, magnetic resonance spectroscopy, EEG, SPECT imaging, sometimes microdialysis, and various other efforts in microscopy. And each one of them will tell you something different about hypoxia or oxidative stress, things like essential metabolites and apoptosis and cell survival. Each one of these things provides its own picture. So no single modality is going to tell you everything there is to know. Bringing them all together is probably going to do that. That becomes a big data problem. An additional element of this is neural connectivity. Neural connectivity is dynamic. It's changing through development, through your midlife, and decays a lot in aging. Just imagine what's going on in the presence of disease or injury. And so looking at things like diffusion weighted imaging allows you to create white matter models of brain connectivity, looking at brain structure. This is an example of looking at white matter fiber connectivity. If you're not familiar with this and you're saying, but Jack, there's nothing white about this image that you're showing me. Anything that in this image, which is colored red, is oriented as a fiber oriented left to right in the brain. Anything that is colored green is oriented anterior to posterior, and anything that's colored blue is oriented inferior to superior. And in various combinations of those colors will produce an image which looks like this. Clearly, each one of these things is a little fiber line representative of a collection of fibers which goes from one area of the brain to the other. So not only can we look at brain function, we can look at brain connectivity. Being able to break these things down and turn them into something that we as data scientists and computational modelers and statisticians and other people with a computational bent can work with is part of the challenge, is taking that data apart. This is something which my group and I have really kind of pushed on. We call these connectograms. You've probably seen these in a variety of different forms. People call them different things, but we call them connectograms. Where the idea is we've broken up the brain into individual chunks based upon a semi-automated parcellation process to break things into gosh, I think it's like 180 something different brain regions. Each one of these little wedges up here represents one lobe of the brain. Everything that's in the right half of this kind of clock face over here is from the right hemisphere. Everything that's in the left side is from the left hemisphere. Each one of these little wedges reflects a lobe, and each one of these little colored stripes here represents that particular region or subregion of that particular lobe. Each one of these different heat maps reflects things like different atlas spaces, for example. So maybe you're familiar with the Distro atlas or the Campbell atlas or the Brodmann's areas, et cetera. Being able to break things down across multiple atlases is actually very important because each of those atlases represents something different about how the brain can be parcellated, either on the basis of gross anatomy, chemoarchitecture, cytoarchitecture, or a number of other factors. We can also look at cortical morphometry. So we can look at things like the volume of that particular area of the brain, its surface area, its thickness, its curvature, various spatial properties of that particular brain region. We can encode that. Likewise, we can look at connectivity metrics such as functional connectivity as well as structural connectivity, looking at degree, participation coefficients, and et cetera, et cetera. And then everything in the middle here is a representation of how that particular region is connected to all other regions. And we've color-coded that on the basis of if you are red, you're in the upper tertile of the distribution of fractional anisotropy, which is a term you're probably all sort of familiar with. If you're green, you're in the middle tertile, and if you're in the lower tertile, you're blue. And we can go and represent these things as this like single image. And so we're compressing a lot of information down into this one picture, which once you get used to looking at these, they become quite attractive and they do make pretty pictures. But it's a way to represent multiple threads of data in one particular image in a snapshot. We've used this methodology to look at something called the claustrum in the brain. So many of you may be familiar with it. If you're not, this was an area that is somewhat mysterious. It was first described by somebody named Felix Vic Desir in 1786. He was the personal physician to Marie Antoinette. And he wrote a textbook where he was the first to kind of showcase and illustrate where the claustrum is in the brain. It's this skinny little region, which is sort of in the shape of a potato chip kind of on end. It's about that thick and makes it very difficult to image. You can kind of see it when you look at anatomical preparations here between the putamen and the insular cortex. It's this little kind of ribbon of gray matter cell bodies here. And this comes from the Rasmussen collection. When I taught at UCLA, I used the Rasmussen collection all the time to teach neuroanatomy. And I was always very pleased to be able to see the claustrum put there. You can kind of see, as I mentioned, it's kind of sandwiched between the extreme and external capsules in the brain, kind of between the putamen and the insular cortex. And it's very interesting because this has a kind of an interesting history, modern history. Is that Francis Crick, yes, that Francis Crick of Crick and Watson fame, he in his elder years began to study things about the brain. He was very interested in consciousness, and he worked with Christoph Koch, who is one of the leaders of the Allen Brain Institute, which is just down the street, to work on this paper. And while they were working on this paper, Francis passed away. And when somebody went into his office and they took a camera in to take a photo of his desk, this paper was sitting on the top of his desk. So he was thinking about what is the function of the claustrum. Christoph worked on this a little while later, and it became published in the Philosophical Transactions of the Royal Society, and really kind of spurred a new interest in what is it that the claustrum does. So anyway, I got very interested in this, and I interested my colleague, Corinna Torgerson. And we went and we used diffusion tensor imaging to try to characterize the claustrum. This is a full brain connectogram looking at all possible fiber pathways in the brain, representing I think there were over 100 subjects that we included in this particular study. If we just isolate the claustrum, which you'll see where it is in a minute, it's this little area which is kind of just down here. We've kind of isolated it down here. What you'll notice is the claustrum is pound for pound, even though it's a small, irregular structure, pound for pound, it's one of the most connected brain regions in the entire brain. It connects to the frontal cortex, connects to the limbic lobe, temporal lobe, bilaterally, all over the place. You'll notice that we've turned off connections to the insula because it would have dominated everything because it's like proximal to it. But this really represents the fact that the claustrum, using large data from diffusion weighted imaging, we were able to go in and identify how this particular structure was connected. This gave us some insight into some of the conclusions of Francis Crick and Christoph Koch. And so we were particularly excited about that. Now, there are a number of different brain resources which are online and they're available to you. You heard maybe a little bit from Russ Poldrack about some of these. And these are really quite a rich collection of resources. These have largely free data sets available to you. Some of them are neuroimaging. Sometimes there's EEG data. If you're lucky, you might get some genetics data as well. These are all free for you. And if you're willing to pursue them, you may have to fill out a web form or get a permission. Some of them are a little more, the gatekeeping is a little more severe. But for the most part, these are no cost or very low cost to you with only a couple of exceptions. And these are great resources for you as you're thinking about hacking on something. So maybe you have your own data, that's wonderful. But if you want some sort of normative samples, these are some that can come to you. And with regards to these, being able to create brain atlases is something that we're often very interested in doing in neuroimaging. That is, we're interested in fitting things to atlases. We're standardizing our brain anatomy so it fits a particular space, or we're very interested in creating new atlases, for example, pediatric atlases or Alzheimer's disease atlases, et cetera. So disease-specific atlases. And there are a number of these which are, we have on our bookshelves, but you'll probably be hearing from my colleague, JB Pauline, about some of the ability to go across some of these digital atlases, identify different subjects who meet particular criteria, and then go out and do a big meta search across different databases to identify where that data is so that you can pull it all down and do a very specific analysis. So again, really kind of driving home the idea that there's just data, it's floating out there and it's waiting for people like you. Now, one of the things that we've talked a little bit about, I think Russ mentioned this, or maybe it was mentioned before, is the Human Connectome Project as one of those particular databases. One of the things which is really interesting about the Human Connectome Project is that it's talking about the sum total of all connectivity of the human brain. If you think about that, though, it's everything in the central nervous system. So it's from about here up, and everything from here down, from your ears downward, is unmapped as part of the Human Connectome Project. And it occurred to my colleague Andre Irimia and I a few years ago that the rest of the connectome is also worth mapping. If you think about it, Parkinson's disease, various other kind of neurodegenerative processes, et cetera, affect the peripheral nervous system. There are a number of different motility disorders. There are a number of different kind of just motoric disorders where people have problems that involve their peripheral nervous system. And so to me, the next frontier of human connectome mapping is mapping everything between your little toe and the top of your head. This will be where you folks come in when that time comes. But if you really think about this really deeply, at every different level of spatial and temporal resolution, there is an individual data science that can happen at that particular resolution, whether it's in subcellular spaces or it's all the way up to regional and whole brain analyses, whether it's in mice, whether it's in humans, whether it's in non-human primates, and whether or not it's involving whatever time or spatial scale that you care to work at. There are a number of these different data science resources which are probably available to you, probably free, and available for you to run your latest, greatest machine learning methodologies on. Now, one of the things about machine learning is you see that I've commented or in my talk title, I've commented this is a science of data. It's not necessarily data science only. There is data that requires a science of that data, not just we're going to go collect data and then we're just going to go do data science on it. It really requires a science of that data, how that data is handled and processed before you even get to the machine learning, the AI that you want to do. Your data has to go through a number of different steps. You'll come with data from a number of different devices. Perhaps you're interested in integrating that data. You'll have to pre-process that data in some way. There'll be some sort of dimensionality reduction because no doubt you've got many more dimensions than you can possibly, realistically manage and model. And that you'll then undergo a number of different kind of parameter estimation methodologies using whatever your AI related tool happens to be. And then as Russ asked us to do is to make sure that we do some model testing. The one thing I would ask of you, and I hope that this isn't too bold of an ask, is as you're thinking about the machine learning that you want to apply, please don't do this, which is start with support vector machines and then systematically work your way through the litany of all the different AI methodologies and say, ooh, look, random forest did the best job. This might be interesting for patient diagnosis. One, that's not telling us anything about the brain. That's telling us everything about those machine learning algorithms. And two, it will not have anything to do with diagnosis. If you try and present that to a clinician, they will ignore you. Really don't do that. I'm a journal editor, and when I see papers like that, immediately reject them. They're not helpful anymore. Be principled about what it is you're going to apply to your data and think deeply before you go through and start creating giant tables of area under the curve statistics, because it's not really going to be helpful for understanding the brain. May be very helpful to understanding your algorithm, but it won't be helpful for understanding the brain. So to wrap up this kind of portion of this, you really want to think about where does data science get its computational inspiration? And it shouldn't be too surprising that it's getting it from the brain. We hear talk of computer vision, of machine learning, of neural networks, of artificial intelligence, of synthetic sentience. And we see examples like this all over YouTube, where cars are trying to teach themselves how to drive, or you have two AI chatbots with little avatars talking to each other and having a reasonably plausible conversation. It shouldn't be too surprising that the brain is trying to use data science to emulate itself. This blows me away. This is a really fascinating thing. And so as you're thinking about this and you're starting to kind of like think about what kind of project you're going to do, and you're going to start writing code and it's going to start pouring out of you, whether you're not using ChatGPT or the latest AI thing to do it, that think about the brain as the source of this. And that not only is the brain kind of the interesting thing to be studying, the data science itself is kind of interesting. So I would argue that data science is also a brain science, and that we're really trying to meet in the middle. And that's why, like, my position at the University of Virginia with being kind of half in psychology and half in data science is really kind of this nice sweet spot for me. But I want you to think about those things as you're considering what projects you're going to be putting together. Now, before I go into the next thing, are there any questions? You should always feel free to interrupt me. Any questions on Zoom? I'm not looking. Okay, I'm going to carry on with this next portion. Okay, neuroimaging databases are somewhat of a success story, believe it or not, in neuroimaging. And I want to provide a brief history. Early on, when I got started, what we would often do is take the collection of local maxima that came out of your statistical analysis, separated by some criteria, and create these tables. We still produce these. You'll see these in any kind of journal article looking at functional imaging. Is a table of local maxima. You're probably all familiar with the TALRAC or MNI coordinate space. So you'd have the coordinate, and then you'd have the statistic, and then it would be that coordinate is localized to a particular brain region, and we say, eureka, the cingulate gyrus is important for whatever. And so, around 2000 or so, some colleagues and myself, we decided that those tables of coordinates were sort of necessary, but they weren't sufficient to be able to describe the underlying data. And that having access to the original data would have been revolutionary, right? And revelatory. So we created a project called the fMRI Data Center with NIH and NSF funding. And the idea was to capture the data that went into peer-reviewed articles that appeared in the literature, and capture that data, package it, anonymize it, bundle it, organize it, and the organizational structure was sort of like a proto-BIDS kind of a structure. If you're familiar with BIDS, we'll be hearing more about that, but sort of organized in that way. And then take those individual studies and supply those to anybody who asks for them, anywhere in the world. And a typical study, for example, from this particular resource, again, keeping in mind this is around 2000 or so, typically had about between 1 and 3 groups of people doing some sort of cognitive neuroscience type task. They were doing a memory task or a language task, visual processing type task. There were about 20 subjects per group. There were usually about 5 runs per subject, 300 volumes per run. And over the course of this, you'd end up getting like 90,000 volumes, 60 gigabytes of raw data, 1.2 million files processed. And then we ended up with hundreds of studies like this. In its day, it was really quite impressive. We had a website and we sent data all around the world. We sent data to the Middle East. We sent data to New Zealand, to Cuba, to Iran, all over the place. We actually had to get special State Department permission to send it to Iran, which was kind of cool. And so we built this and we were really excited about it and had nothing but good intentions. But of course, it was sort of before its time, maybe. Because what we would do, before I get to that next piece, what we would do is we actually burn the data to DVD and physically mail it to people. Now, maybe in today's technology, right, you think, oh, that's kind of silly. But keep in mind how Netflix used to work is you would go and you'd ask and they would mail you a DVD with your movie on it, and then you would mail the DVD back, right? So at the time, this was the kind of technology of the day. And so we would burn the DVDs with that particular study and then mail it to people, again, all around the world. And this is an example of what it looked like. This is work we did at Dartmouth College, since the name. So what's kind of interesting about this is we started this process and thought, wow, we're doing something really good for the community. but the community lost its freaking mind about this. So articles started coming out in Science and Nature and in PNAS saying that, oh, prospects of data sharing give brain mappers a headache. People said, whose scans are these anyway? And why would we want to send these to those knuckleheads at Dartmouth College? People came out with articles in Nature talking about the debate over fMRI data sharing and whether or not we didn't understand what the bold response was. It was too early to start sharing data. We needed to wait until we understood the bold response. Last time I checked, we still don't understand the bold response, right? And people started coming up with these like litanies of different criteria that you had to kind of subscribe to in order to be a good data sharer. Even the Organization for Human Brain Mapping, of which I am a member, and many of you are probably as well, came out with a treatise on what it meant to share neuroimaging data and what criteria databases should have. And people came out with impassioned arguments about databases being good, bad, and ugly. And so it wasn't too surprising to get a sense of how people thought of us. They considered us part of the ugly group. That wasn't the way we did it, or the way they would encourage to do it. So I was a very junior faculty when these articles started coming out. I was just like, oh my God, this is like, what have we gotten ourselves into? We persevered with this and continued to do it. And of those hundreds of studies, we sent data to people all around the world, had multiple copies of this, floating around, was really quite ahead of its time. And due to the wisdom of the National Institutes of Health and the NIH, they kind of got bored with it and moved on, and it basically ran out of funding. A number of people left, and so the database kind of ran its course. People often ask me, was the fMRI Data Center a failure? I don't believe it was a failure at all. I think actually it was an interesting experiment that actually opened the doors for all of those databases we just talked about a moment ago. All of their history can be traced back to our experience with the fMRI Data Center, dealing with things like anonymization, dealing with things like how do you organize all those files? How do you disseminate that data out to people in a way that's convenient and could, not overwhelm somebody's hard disk, keeping in mind that in the early 2000s, the average size of an fMRI study was about the average size of somebody's hard disk. It was a lot of data. So that by sending it out on media, it was kind of an interesting way to do it. fMRI in the beginning was a lot about task activation. So this, and I'm sure that JB Pauline and others are old enough to remember when this Science cover came out from Jack Beliveau and colleagues from the Massachusetts General Hospital. They were the first to kind of publish what is more or less related to a modern fMRI experiment, where you're flickering lights at people and you are then looking at the wave pattern of the on-off cycling of that particular stimulus pattern, and you're then looking at the pattern of activation. And so all of those initial studies were either like flickering lights or wiggling of fingers. When I was about where you guys are now as a postdoc at the NIH, I did a lot of finger wiggling experiments where you lay in the scanner and people would do this and then they would stop and then they would do this and then they would stop. And it was that kind of square wave pattern that would give rise to your pattern of activation. Then people started to get a little more nuanced. So looking at things like, for example, pictures of faces, pictures of houses, and pictures of chairs. This is some work that was done by Alameda Shai, working with Jim Haxby and Leslie Ungerleider at the NIH, where they're really looking at and kind of helping to define that thing we now know as the fusiform face area. And so these initial studies were able to help us define that, look at these differential patterns and get an idea of kind of stimulus-specific patterns of activation. More recently, things where we're using multivariate methodologies to look at the subspace that these data sit in is been the way forward. So people look at eigenimage type things with looking at principal component analysis or independent components analysis. Everybody who's familiar with the resting states default mode pattern of activity, that's where that comes from, is analysis of these multivariate patterns to be able to pull out these areas which are correlated with each other and co-activate. And the default mode is one of them. Doing studies of the brain at rest sort of has almost overwhelmed those initial task-based activities. If you go to meetings like OHBM or various other Society for Cognitive Neuroscience, for example, there are a number of different resting state studies now where there's no task at all. People lay in the scanner, you're just measuring their brain and you're asking, what is it about this that characterizes their default mode network? This was a study that was done by Malia Mason. I was involved in this. Again, this was back at my Dartmouth College days, studying the brain at rest, where we realized that the brain is never truly at rest. It's always doing something unless it's dead. And that the brain is always in this state of what we called stimulus-independent thought. It is that resting state mode of activity. When you're laying in the scanner, you're not thinking about something. You're thinking about, oh, I got to remember to buy milk on the way home, or oh, I've got a big test coming up, or oh, I need to call my mom, or whatever it is. Or you're thinking, gosh, this is a very loud scanner environment. You're thinking about something, but it's not driven by an external stimulus. It's driven by your internal state. And this appears to be something which is related to the tendency for people to daydream or not. So we went through and did a study where we asked people about their daydreaming kind of, I don't know, habits. And we found people who were particularly good at daydreaming. That is engaging their default mode activity. And then we'd ask them to do a task. They would disengage from that, but then they would immediately go back into this daydreaming capability. So we went through and we looked at this and ended up demonstrating that this notion of stimulus-independent thought just appears to be this natural property of the brain, that brains just do this. And it was quite exciting kind of thing to look at. White matter fiber mapping is kind of the next thing I wanted to touch on with this. And this really gave rise to things like connectivity analytics, being able to look at brains as nodes, collections of nodes and edges, and analyzing their properties, looking at things like their small world properties, their kind of community structure, et cetera. And the notion of this connectivity was really the basis for the Human Connectome project. In the past, we've at other events that Ariel has run, we've heard from folks who are deeply involved in the Human Connectome project, and that is just such an impressive effort. I won't attempt to summarize it here, but to say that looking at structural connectivity really allowed us and my team when I was at UCLA to look at historical cases of connectivity that were really quite interesting. So not real data, if you will, from real people, but to simulate data in somebody who was dead, but who had a very famous brain injury. Who's familiar with the case of Phineas Gage? You're all familiar with it. It's always in the first 24 pages of any Psych 101 textbook, right? So Phineas Gage was a railroad worker. For those of you not familiar, railroad worker, and he was demonstrating to his team how you're supposed to blast rock to make way for a railroad trestle. And so he was ordinarily you do this by digging a pilot hole, you pour black powder into the hole, you put in a fuse, you put in some sand, and you tamp the whole thing down with a tamping iron, and then you light the fuse, you run like hell, the rock explodes, and then you go clean up the rock, and you continually march down the railroad track to produce to flatten the land. And in Vermont, there's a number of basically it's all granite. So this is a very lengthy process of blowing up little patches of rock and then kind of marching down the thing. And so Phineas was in charge of this group. He was demonstrating this. And at some point during this process, somebody forgot to pour in the sand. And if you take a large 1 meter tall piece of iron shaped like a javelin and drop it into a hole of granite, what's going to happen? You're going to get a spark. Right? That spark's going to ignite the black powder, and that javelin-shaped thing called a tamping iron is going to come rocketing out of that hole, and it did in this case. And it shot up underneath Phineas Gage's zygomatic arch, went behind his eyeball, and shot straight and completely out of the top of his head. As you can see in these little images up here. This is the hole that was left. Here is kind of staring, let's see, yes, this is staring up the hole, so along the path of the tamping iron. And here you can kind of see the crack that it left in his frontal bone there. On that little video I showed you, can I do that again? Try that again. This is an illustration of the skull and how that tamping iron would have flown through his brain, and how the white matter fiber pathways, as pooled over multiple different subjects drawn from a big database, might have actually looked. From this, we can go and do all sorts of calculations on what would have been the effect of a tamping iron passing through a highly connected set of brain regions and how that might have affected the network properties of that particular graph. Without going into too much detail, we can go and create a little connectogram, and we can show which were the fiber pathways which were transected or affected by the passage of that tamping iron through Phineas Gage's brain. And this is kind of the extent of this. One thing which is really kind of interesting is that previously people had said that the tamping iron crossed the midline. In our analysis, it did not cross the midline. In fact, it was largely a left hemispheric problem. However, it did affect fibers as part of the genu of the corpus callosum, which would have crossed from one hemisphere to another. Also, in as much as it was a gray matter injury, it was also a white matter injury, in the sense of it was about 5% of Phineas Gage's gray matter volume was affected, but 11% of his white matter volume was affected. And so we thought this was very interesting, gave a completely different picture to how one would interpret the case of Phineas Gage. Being able to deal with data sets like this often requires you to build a workflow. We just heard from Russ to talk about how really for every different study, there's probably every different workflow. And there is no standard workflow. In fact, whenever people ask me about, oh, what's your workflow? I'm like always a little nervous because there's always a different workflow and you're always tweaking it and changing it and adding bits to it. And they actually become quite diabolical. And in fact, when I was at UCLA and at USC, we used a tool called Lonnie Pipeline, where we could go and create these very bespoke sets of workflows that would do anything to your data. And it would utilize software tools such as AFNI, FreeSurfer, FSL, many of the types of tools that you're probably familiar with. Anything that we could code in Python, compile in MATLAB, or run as C code executables, we could then bolt in as a module to play with all of those other tools as well and create these Franken workflows that did just about anything to our data, and often did. At the same time, we were organizing data in BIDS format, was still at that time a little bit new, but now is fully mature, being able to draw from not only our local storage, but from cloud storage, and to be able to take advantage of other different things within this framework to inform and color our interpretation of those data, which is a very important element, often missing when one is supplying or looking at data from a machine learning point of view. One of the things which is kind of interesting about all of that is we talked a little bit about statistical power with Russ this morning and those different workflows is John Ioannidis from Stanford published this article. And again, Russ mentioned this as well about why most published research findings are false. Neuroimaging has been particularly problematic in this space. John points out that of the most highly cited 1,000 or so neuroimaging studies, they had a sample size of 12. Not sample sizes of 1,200 or 12,000, but 12. Interesting story. In the early, how many of you are familiar with statistical parametric mapping? Software tool, yeah? So in the early days of statistical parametric mapping software, it was given away free, but the sample size of n equals 6 was hard-coded into the code. You could not do a study of other than n equals 6. So if you look back in the 1990s, early 2000s, you will find a kind of a preponderance of sample sizes of n equals 6. And that's the reason, because you couldn't do another one because it was hard coded. It took some bright sparks to go in and find the places where it was hard coded and change that to a variable so that you could have other sample sizes than that. John also points out that a sample of about 300 studies published between 2017 and 2018 had sample sizes a little bit larger than that, and sample sizes increased about 0.7 participants per year over that time. It's really kind of remarkable that a number of these things that we've now based a number of different fundamental neuroscientific ideas about were based on these very, very poor sample sizes. When Russ was talking about this kind of pattern in a graph of this kind of like early studies over-reporting the effect sizes, and that tends to get smaller over time. I added this really quickly because this is the very first article I ever published as a scientist from my PhD related work. This is called Ventricular Enlargement in Schizophrenia, and we did a meta-analysis of studies of something called the ventricular brain ratio, which was a metric where in imaging data, you take the image of the brain with the largest depiction of the lateral ventricles in schizophrenics. You kind of outline those and get a calculation of the area, as well as the area of the table of the brain at that particular slice. So you take the ratio of the ventricles, the ratio of the whole brain, and you multiply by 100. That's the ventricular brain ratio. And when people reported this, the earliest studies reported these very large effect sizes. But as people got better at statistical kind of statistically controlling for things, adding more subjects. One of the other things that people did was, in order to get a control sample, they would go and they'd find like headache cases. And so they would go through the kind of file drawer of a radiologist and identify people who weren't schizophrenic, but maybe had come in to have their brain scanned because they had a headache. And they would systematically go through those files, hold the x-rays up to the light and go, oh my God, look at the size of those ventricles. That person can't be normal. And so they were systematically trimming off the upper tail of the otherwise normative distribution of the ventricular brain ratio. And so they were overinflating the size of the effect. In addition to having relatively low sample sizes. We went through and repeated this analysis that was originally published in the British Journal of Psychiatry, but we reanalyzed it 20 years after. We reanalyzed that data and we added a number of other ventricular brain ratio studies that had occurred in that time. And we found exactly the same effect, as well as looking at things like funnel plots like this one to indicate that those smaller sample size studies have enormous potential variance in them. And so this kind of drives home some of the point that your sample size and statistical power are vital elements in your kind of thinking about how you're going to do any kind of analysis at any kind of level. A number of other things have cropped up in the intervening years. There was an article published by Anders Eklund working with Tom Nichols and others, which they called cluster failure, why fMRI inferences of spatial extent have inflated false positive rates. And what they had noticed, amongst other things, was there was a 15-year-old bug in some classic software as part of the AFNI distribution that actually got the calculations. There was injected a bias in those calculations, which underestimated the severity of the multiplicity correction and overestimated the significance. The press got a hold of this, and there was a mini little storm about how half of all fMRI studies are wrong. It was probably not quite that bad, but it certainly did depend. A lot of people depended on the value of those statistics in what they reported in their peer-reviewed articles. Now, you might think, okay, that's a one-off. That probably doesn't happen all that much. Actually, it's happened before that too. Back in 1995, there was a test as part of SPM, again, not to pick on SPM, it just is another good example, but where the number of activated voxels above some level of expectation in positron emission tomographic analysis using SPM was assessed using a chi-square statistic. And it was assessed with a single degree of freedom, but very often the chi-square values themselves were reported in the hundreds. So somebody would report a chi-square with 1 degree of freedom of a value of like 300 or something. Well, if you know anything about a chi-square distribution, you know that, wow, 300 with a single degree of freedom is like, that's impossible. Well, it turns out it was impossible because they had used the wrong methodology for calculating the chi-square. They actually used a different methodology, which wasn't appropriate for the kind of data that they had. They also did not account for image smoothing, which is that tendency for the data to be autocorrelated. So the neighboring voxels tend to be, correlated with their other neighbors. And if you did this, you'd actually go and adjust for the proper calculation and incorporate the smoothing. It would actually give appropriate distributional properties. But in the next issue of SPM, that chi-square statistic conveniently vanished. Quietly vanished, and there was no more mention of it. This was after I and some other colleagues had written a letter to the editor pointing this problem out. Maybe I had something to do with it. or they just cycled it off. Now, one of the other things which you'll often hear about is the famous salmon study. How many people are familiar with the dead fish salmon study? So these were colleagues of mine when I was at Dartmouth College. Uh-oh, I did it again. There we go. Loose connection. And what is the funny backstory about this is that they were often in like the wee hours of the night, they were testing their paradigms, and they would often go to the grocery store and they would buy watermelon or celery or, you know, bananas or something, and put it in the scanner and use it as sort of a dummy for them to test their analyses. And then, you know, looking at cross-sectional anatomy of bananas is maybe a little amusing, right? So that anything that could go in the scanner, they'd put in the scanner. And then Craig Bennett, who was the leader of this study, had gone to the store and bought an Atlantic salmon. He was going to have a salmon cook, and he bought the salmon and said, oh, well, let's put the salmon in there. And then he did his analysis. And sure enough, there was an activation. You can kind of, it's a little bit hard to tell, but if you look very, very closely, in the brain, or where the brain is supposed to be, of this dead Atlantic salmon, there was a pattern of activation, according to, with respect to his task. And so he kind of wrote this up as a submission to the conference OHBM and submitted it. This caused fits amongst the people who go and evaluate the poster submissions for OHBM. And there was a huge, big controversy about, was this a joke? You know, what's going on here? And in fact, it was, I think, Russ actually defended this, said, no, this is actually a good example of like why we need to be more careful about how we deal with multiple comparisons and what we interpret as a pattern of brain activation. Interestingly, Craig Bennett and colleagues, they wrote this up in the Journal of Serendipitous and Unexpected Results. And as a consequence of this, they were the 2012 Ig Nobel Prize winners. For many of you are familiar with that. So it's, again, very interesting little story. In doing meta-analysis, Russ pointed this out as well. People don't tend to publish not significant findings or findings which are the other way around. People tend to publish findings which support their hypotheses. So we end up with something called a publication bias very often. And you can go in and measure this as we did a few years ago, where if you plot out the distribution, or sorry, the relationship of the size of the statistical effect in terms of either a Cohen's d, or you can convert things to a correlational like metric of effect size against sample size, you can see kind of a pattern which looks like this, where you get this big cluster of relatively large effect sizes for relatively small sample sizes. And this is largely because if you go and look at something like this, people will go and they'll do an analysis and they'll go, oh my God, look, it's significant, and they'll rush it to publication. And one thing that we found in a subsequent analysis of this is that the size of the statistical effect is often inversely proportional to the number of authors on the paper. We thought this was really interesting, right? Is that the more you can imagine that you and your buddy get together and you're doing an analysis and you get your 6 subjects, you find some huge big effect and you go, woohoo, we're going to science. And there's no one there to kind of like sanity check you, right? And so the more authors you have, the more eyes are on that and the more people asking questions. Probably not too surprising that there's a little bit of a reality check in kind of having these large scale teams. But then other people more recently, however, have indicated that those small sample sizes are probably not sufficient, and we need to be very, very careful about them. And this was an article which came from Merrick colleagues talking about, and Russ touched on this as well, talking about brain-wide association studies requiring thousands and thousands of individuals. For any one of you to try to collect that amount of data is next to impossible. You don't, if you were to start today, it would take you your career and probably longer to be able to collect that kind of data. This is a justification for having these types of archives which we can bring together, which we can utilize collectively, such as, you know, Human Connectome Project, et cetera. Here's just a sample of these. This is from Russ Poldrack's group at Stanford, the Open Neuro effort. You're probably, many of you, familiar with that. Likewise, there's the Alzheimer's Disease Neuroimaging Initiative. For anybody who's interested in Alzheimer's disease, out of this archive have come hundreds of research articles, thousands of conference presentations and posters. It is one of the big success stories in human neuroimaging as related to neurological disease. The Federal Interagency Traumatic Brain Injury Research resource is available for anybody who's interested in traumatic brain injury as a condition of funding, excuse me, for your TBI related research. You are through the NIH, you are obliged to submit your data to the Federal Interagency Traumatic Brain Injury Research Resource. So there's a lot of data in there. Oops. And one of the things that is particularly interesting is the effort of my colleague Paul Thompson from the University of Southern California called the Enigma Project, which is enhancing neuroimaging genetics through meta-analysis. This is a slide that is woefully out of date. At one time, they had 70,000 plus data sets with investigators from all over the world, broken down into different disease subclasses in terms of ADHD, autism, TBI, bipolar disorder, amongst others. This is, again, there's multiple different working groups on over 20 different disorders, 39 countries and counting, number of different scientists, and they have the largest GWAS studies of the brain published in journals like Nature. Very often, when you look at studies like this, you'll see that these are these large scale co-author efforts where the number of authors is very often more than the number of words in the abstract. These are kind of big data science related neuroimaging efforts, but they seem to sweat Nature and Science publications. For those of you who are interested, it's really kind of low cost to get involved with them, and it may be something very interesting because they provide the processed data to you so that you don't have to do all that processing. You can dive mostly right into where you'd like to with some of your machine learning. Of course, those are the data which are available. There's also the dark data. That's the data that the experiment didn't work out, the postdoc left, and that data is sitting around, and it may be in places like various lab hard disks, variably accessible file structures, EHR, closed databases, a number of other different places. This is often referred to as the dark data, that data that somebody paid for but hasn't really seen the light of day. Being able to go through and identify that data and bring it to bring it to light is kind of an important area in order to be fair. Many of you are familiar with those FAIR principles. That is that data should be findable. It should be possible to locate that resource easily on the web, that it's accessible, that there should be few technical barriers to get access to it. It should be interoperable. It should be in a format that allows you to interoperate with various tools and have that data play well with other resources. It should be reusable, that there's no real restrictions on having purpose beyond its original intention or audience. And I always say that FAIR has a silent E on the end, and that E is for education. If we're not gathering these data and making them available to students and to postdocs and to trainees, maybe like yourselves, then we really are missing an opportunity to be able to do something for our community. So that silent E is important. Russ mentioned a little bit about this best practices in data analysis and sharing in neuroimaging. This was an effort that back when I was part of the main leadership in OHBM, we collectively asked Tom Nichols, who some of you are familiar with or have collaborated with, who's now at Oxford University, to lead this effort, and they did so and produce what's called the COBIDAS recommendations. And these are recommendations on how one should report the findings of their particular research using neuroimaging, what was done to the data, how it was analyzed, basically providing the information needed for somebody else to independently analyze that same data and get the same result. COBIDAS is going through a kind of new revision now. Probably in about a year or so, there'll be a COBIDAS 2.0, which will extend this idea as we know more about kind of modern methods of analysis and data collection, et cetera. Now, many of you may be kind of wondering about what happened to that fMRI data center effort that I mentioned to you earlier. Like, what happened to all that data? Well, the servers that it was on crashed moons ago, but in my basement at my home in Keswick, Virginia, I have tubs and tubs and tubs full of fMRI data center DVDs. Here's just an example of a picture I took the other day. I've got these giant sleeves of DVDs. This is the only residue of the fMRI data center left after all that effort. So it's in my basement, basically, if you want to know where to find it. So really, human neuroimaging is this kind of modern success story. I mean, for many of you, you think that all this stuff just existed for all time. There was actually a huge, big kind of set of growing pains that the community had to go through before it realized that sharing was something which was beneficial to us all, because it wasn't always the case. And people had to either be required to do it, so there was kind of a carrot or a stick approach. You'd depend upon people being benevolent, or you'd make a condition of publication, or sorry, a condition of publication or of funding to be able to get people to share their data. And eventually they started to do it. The neuroimaging data types are so widely varying and so covering clinical disorders and cognitive dimensions, and how you process that data is also different. Being able to share that original data is really important, to go back to the original data that was used for that study and to be able to do not only replicate that particular study, but then to take that replication and kick it up a notch, right? Do something more to it and find something else which is present in that data. Organizing that data is a challenge, but it's not impossible. And really, it's through the sustained attention on the importance of sharing that we're all going to benefit of this. Really, it's community standards like BIDS and fMRI prep and other things that we've adopted really help to make that data more useful for the maximum number of people. And when we're looking particularly in clinical disorders, we want to be able to make sure that we've maximized as much as we can. And so really that data science applied to brain science effort is really all the better for all of this sharing. So before I go on to this next little bit, are there any questions about that? Yes, sir.
Wondering what the qualms were to deliver to your critics of the fMRI data, and if you think they still would have similar concerns about the data sharing again.
People were concerned about being scooped. They didn't want to give away the Nobel Prize that they thought was present in their data set. Very often people would say, oh, my experiment is so complicated, I couldn't possibly explain it to you. And when you pointed out that that isn't really science, they kind of got a little chip on their shoulder. People were just very nervous about giving away data. They were concerned about anonymization. They were concerned about, well, what happens if there's a secondary finding? People are very concerned about those things. We seem to have gotten over most of those things now, although there probably still is a little bit of reticence about giving away a complete data set before you fully had an opportunity to mine it for all it's worth. But that was probably the biggest things then. Also, the ability to share data. The internet, as with its speed and capacity in 2000, was a lot different than what we have now, where we can upload a whole stream of DICOM files and have it kind of get to where its destination. Wasn't always very reliable in 2000. So people would literally take the hard disks out of their computers and mail us the whole hard drive. So the delivery of the data was always a challenge. Any other questions? Yes, sir.
Where you had with all those studies and information from it, isn't it, if it's not already, would it be practical or even possible to upload that into a modern form that we could see it today?
I wish. Well, yes, in principle, if I had a DVD reader, which I think are becoming increasingly rare, and an army of undergraduates to put disks into drives and offload that data, which I don't have, which would cost money. And so, technically speaking, yes, that could be done. Practically speaking, it would be a lot of work. But great question. Because even though that data is old, it's part of our historical record. And I think that means it has value to us from a historical point of view of how we did studies back then and what our challenges were and how far we have come. It's a measure of how far we have come. So, all right. I wanted to talk about how we're using data to create models and coming back again. And then what I want to kind of finish with, I'm hoping I'm going to be able to do this fairly quickly, is how we're doing some stuff now with simulating kind of neural networks using kind of digital twins, inspired by all this analysis that we've done. So many of you are familiar with the work of Ramon E. Cajal, and looking at data, if you will, in the form of slides and looking at them under microscopes and being able to characterize what you see and be able to make kind of inferences about, say, in this case, if you look in here, you see all these little arrows embedded in here, indicative of the flow of neural impulses through, in this case, the hippocampus. And who doesn't love the hippocampus, right? Being able to look at white matter fiber pathways in the brain is something that is still relatively kind of unfamiliar to us in neuroimaging. Not to say that we're not doing it with looking at diffusion weighted imaging. We can certainly get out those pathways, but modeling signal transduction along those pathways, that's something that we haven't really thought that deeply about. We kind of sort of do some hand waving to think about it, but we haven't really said something specific about how this white stuff sandwiched in between layers of between the levels of cortex is actually conducting information. So we started to think about this a little bit and to come up with some ideas which we think are going to be really fruitful. So many of you are kind of familiar with looking at diffusion tensor imaging and modeling of these different diffusion directions as a tensor or set of tensors, and breaking things down into an eigenvalue structure and getting out kind of a magnitude as well as a direction in terms of the matrix of water molecule diffusivity. That's super important because you can go and look at fractional anisotropy, for example. You can compare neonates with 2-year-olds, with adults, and you can look at not only looking at the DTI data, but you can look at other metrics as well, fractional anisotropy, mean diffusivity, et cetera. And of course, you can do all of the white matter tractography, which we kind of described before, and then do some kind of calculations on those particular patterns of connectivity. However, there's a lot more going on than just tensors, and it's looking at brain microstructure in white matter fiber structure is actually a lot more complicated. There's a lot more going on. This was an article from Stenson Co. Sorry, this little thing's in my way, so I'm not going to pronounce his name properly. Beg your pardon. But at any rate, looking at the different things that are going on microscopically at each different kind of pattern here. So you've got all neurons in their cell bodies. You've got spiky neurons. You've got astrocytes. You've got all sorts of different things, all within the same kind of section of neuroanatomy. So there's a lot going on, and we'd like to use neuroimaging to be able to get a better idea of how to characterize the different compartments of water molecule diffusivity more than just from a tensor-based approach. So my colleague Ben Newman and I have started looking at this 3-tissue constrained spherical deconvolution model to break things up into intracellular anisotropic, intracellular isotropic, and extracellular water based compartments. And so we can model up to 5 fiber bundles per voxel with interaxonal volume and orientation for each one. And this really works well for pathological samples as well as for normal samples. And we can get out a number of different metrics based on this, not only looking at if stuff is intracellular and extracellular, but then we can combine those in creative ways. And let me show you a little bit more about how we've done this. So this is looking at a kind of a free water component, a kind of a white matter component, and a gray matter component, looking at different B values in here, creating kind of overall signals and being able to create these little spherical deconvolved kind of little objects that are like on the right there. We normalize these so they have a sum that equals to 1. And from those, we're able to actually make a few more calculations that takes like a tensor map like this and do some improvement on it, which really kind of sharpens it, right? It's like getting LASIK surgery for your diffusion weighted imaging analysis. So this is kind of cool. We can look at these 3 different components in things like aging, for example, where that extracellular water component tends to increase as you age. The intracellular component kind of tends to follow this little kind of negative sigmoid kind of thing. And here you have this kind of parabolic arch, if you will, for the intracellular anisotropic component. And you can go and create little profiles of this to kind of show you how these different things change with age, et cetera. We also were very interested in what happens in demyelinating diseases or diseases of neuronal architecture, such as Parkinson's disease. Parkinson's disease has been implicated in having alterations to white matter. As you know, around white matter, or the white matter around an axon helps to improve its efficiency and speed of transmission. And so we were very interested in looking at this. There are a number of different articles which kind of claim this. And we do this by taking those measurements and doing a little bit more magic to create something called the G ratio, which is a measure of the degree of the axonal diameter as a ratio to the degree of the myelinated diameter. So if you can imagine like having two consecutive spheres, much as you've got here, or two consecutive cylinders, basically, where you have a inner diameter here to this little d component, which is the measure of the axonal diameter, to the ratio of the myelinated diameter, you get G. And from that, based upon, which I'll get into a little bit more later, you can make measurements of conduction velocity, or at least a value which is proportional to it. And then by using this, you can go and do a couple of calculations. And this is how you convert G over here into a value, this theta parameter, which is related to conduction velocity. It's not direct, there's no units to it, but it's proportional to it. And with a little bit of a normalization factor, we can go and standardize this curve as you vary G, you can get it to peak at 1 by kind of normalizing it a little bit. But you can see the shape of this is kind of interesting, right? Is as you get less myelin relative to the axonal diameter, the actual velocity falls off quite precipitously, right? Whereas as you get more myelin relative to the axonal diameter, and that also affects G, is that it actually falls off more gradually. So this is kind of interesting, and we're able to go and make measurements of this at every voxel in the brain and get a value which is right around the value of 0.6. So it turns out that it's actually 1 over the square root of E, and that is born out of the equation itself. But it's kind of interesting to be able to go and look at measurements of this at every location in the brain, and then to ask how would signals that are or fiber pathways which are traveling through those particular voxels, where you have that, how might they be affected in something like Parkinson's disease? And so we went in and looked at this in some detail. Here's kind of a bad depiction of our workflow, all the different things that we take into account here. So not only are we looking at the diffusion MRI, we're looking at T1, T2 ratio as a measure of myelination density. And with a little bit more magic, we can go and create those G ratios. And with that equation I showed you before, get out a value which is related to conduction velocity. And then in looking at our Parkinson's disease sample, we had a number of different subjects. There were 25 so far in our sample, and the G ratio has a significant positive association with years after the PD diagnosis in bilateral cortical spinal tracts, which might be expected. And most cognitively significant metrics so far were motor-based metrics, and did not survive, but they didn't, but due to the small sample size, didn't survive multiple comparisons. And there seemed to be something that showed some promise with the VO2 max and how oxygen was utilized. Just kind of carrying this idea forward, even though it wasn't quite statistically significant, as we were seeing different areas, particularly in bilateral hippocampus, caudate, and right amygdala, looking at relationships with, again, caudate and thalamus, as well as bilateral hippocampus and amygdala in relation to memory, and is there another one? Global cognitive score in these Parkinson's disease patients. We've also started looking at this longitudinally. We've got 20 subjects so far, and we're seeing these kind of areas that are related to subcortical regions and cholinergic basal ganglia with a lot of these metrics of interest. So this is something that we're pursuing a lot. The idea here, though, is to actually go back to something that's fundamental in terms of neuroanatomy, something that's fundamental in terms of electrophysiology, related to these neuroimaging metrics that we can extract. It would be one thing to go say, oh, I'm just going to take all my data and pour it into some data, machine learning thing and have it spit out some answer. But it's unprincipled, right? It's actually much nicer to have some sort of an idea and a hypothesis you'd like to test, and then go forth and do it. That's just some more results I'll kind of skip over, but really it's bringing these data from this idea of we've got the data to creating a model, something that we would be very interested in looking at, conduction velocity, which is something of a unique thing that we have not really done that much in neuroimaging, except for various specialized forms of medical imaging. And so this was something that we pursued. Now, to kind of bring this all home, we started taking some of these ideas and bringing them into the scope of how we can simulate little neural systems using some of these kind of ideas that we've extracted from neuroimaging data. Many of you are probably familiar with, a lot of foundational neuroscience, looking at different cell types at microscopic level, and knowing about 7 layers of cortex and how we talked about myelination and G ratios and whatnot. and white matter fiber tractography, we kind of became inspired by this. And a student, Ethan Nelson, who's working with me at the School of Data Science at UVA, has become very interested in this idea of how we can kind of combine some of these ideas to create these little things, which I'll describe in a second. We've also looked at autism with this. Basically, starting from the premise that you have axons that connect different regions of the brain. Those axons have these various properties. One, you have these fiber bundles, you have kind of these myelin sheaths that wrap around the axons to help control the speed of which at which neural signals travel from one end of an axon to another. Can we leverage that? Can we take that idea and kind of build upon it in some sort of a clever way? Well, we thought deeply about this, and a lot of foundational neuroscience is built around what's called the cable equation. The cable equation is one of those fundamental things. You'll find it in like many kind of neuroscience textbooks. It is largely the work of Hodgkin and Huxley. Many of you are probably familiar with those names, who, along with John Eccles, won the Nobel Prize for thinking deeply about how signals transduce, looking in particular at giant squid axons. And they came up with this equation, which is this partial differential equation in both space and time of how signals move through the medium of the giant squid axon. And they were able to define the action potential based upon this kind of work. One thing which is kind of interesting is, for those of you who are engineers, particularly those who like to work with breadboards, it's possible to actually go and emulate this using this kind of circuit model, and you can do it very simply with a circuit board. The idea is that you have this kind of outside area of extracellular fluid. You have kind of an intracellular inside component, and it's these little transistor kind of models actually radiate outwards and help to describe that system. It turns out what's kind of interesting about the cable equation is actually it's a simplification of something which is called the telegrapher's equation. The telegrapher's equation was actually described by James Clerk Maxwell sometime in the kind of mid to late 1800s. It's actually the sum total of all of the Maxwell's equations, and was actually used by Lord Kelvin to describe the transmission problems that were happening across in transatlantic cables from England to New York, for example, and how they were being distorted. And Oliver Heaviside, who's actually responsible for the current format that we see of Maxwell's equations, also took this on, and he ended up solving some of the distortion problems that existed in telegraphers communications, where you're using this little key device, and you've probably seen this in old-timey movies where you're going da 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 da, you know, we're the SS Titanic, we're sinking, da 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 da, right? And they would use cables like this to send signals from England to New York, for example. And here's kind of a collection of what those look like. What would happen is that this equation, this guy, describes a pattern of an impulse, which is given at time kind of t equals 0, and how it modulates over time and over distance. And that Hodgkin and Huxley took this idea and turned it into the cable equation, which is the equation that we find on all of our neuroanatomy textbooks. Interestingly enough, William Albert Hugh Rushton was a colleague of Hodgkin and Huxley's at Cambridge. And he's the one who came up with this particular equation. So we refer to this as Rushton's equation that gives this particular pattern that we've talked about, where when you, as you're looking at myelinated axons, this gives you some insight into how quickly signals can transduce down those particular axons. One of the things that my colleague Ian kind of noticed was we have a number of different architectures for various neural networks that we apply. And they all kind of tend to look the same, but the spatial arrangements of the nodes and the edges really don't matter so much in these depictions. And also the speed of which kind of signals move from one node to another in a computer happens almost instantaneously. We actually don't care how long it takes in order for our learning systems to be able to do what they do. So he was interested in what happens if we take kind of a neural system here, neural network, and we start to deform it. And we start to kind of like spread these things out, fan them out. What if we turn this whole thing into kind of this circle? What if we turned it into a sphere so that we actually have a cortex and we have a white matter, you know, set of a white matter structure? And what if each of these connections has some myelination to it, which has a G ratio, which we can use to kind of put a governor on the speed of transmission? And we can actually use this three-dimensional object using the telegrapher's equations to help describe how signals move through this particular digital object and learn, which we thought was pretty cool. It's like a digital twin. So we were inspired by this three-dimensional structure of it being kind of a ball with nodes all over the outside of it, maybe multiple layers to it, and then white matter fiber pathways connecting the various components of it, inspired by neural organoid biotechnology. Many of you may be familiar with this. You can now, in a petri dish, you can grow these little neural organoids based upon looking at pluripotent stem cells, and with a little bit of magic over here, you can go and create these little mini brains, and you can create multiple of them, put them in a petri dish as kind of depicted here. They're really popular in drug discovery and seeing how neurons might respond to exposure to different drugs, et cetera. So we referred to our things as digital neural organoids or diginoids, or if you're really lazy, you could just call it a noid, right? And these are a three-dimensional object, which is an arrangement of cells that has one or more shells or cortical layers, if you will. So it produces a cortex. Input nodes are on the cells in the outermost shell, and the hidden layers or the internal shells are the kind of the hidden layer components of this. And then you have these kind of central output node ganglia, which are where all the signals get collected and where you actually kind of do a read, if you will, of how the signal is being propagated through the system. There are a number of other properties which we think are pretty cool about this, and it's like a little digital twin of a neural organoid. And we're very interested in seeing if we can teach these things to learn. And the short answer is yes, you can, which is really kind of cool. That signal transduction, one has to be thinking very carefully about this. The speed of that transmission is governed by our little equation here, and we have to very judiciously look at the boundary conditions. For those of you who solved a number of partial differential equations in calculus 101, this is the kind of equation that you'd come up with, very judiciously selecting your boundary conditions to make sure that the signals have the right properties, et cetera. So we've played around with that a lot. This is kind of an example of a very simple one, which we've exploded slightly so that you can see the white matter fiber pathways, and I'm almost done, Ariel. So this is what this kind of looks like, where the nodes, which are yellow, or sorry, are orange, and the fiber lines are connected as the sum total of all the neural voltages which are passing through those particular nodes. We can then go and do a variety of different network statistics on those. Here's another example of where it's there's now 50 different inputs, and we can see how the different time courses for each of those inputs changes here, and how the kind of community structure, if you will, of the particular system itself changes. So we're pretty excited about this. You can do all your digital network or your network analytics, looking at between the centrality and looking at a variety of different other metrics as well. And so this may be a very interesting resource as a in silico learning object. That is a little mini brain, a little neural network that we've constructed to be three-dimensional that we can use to learn. What we're doing right now, and I'll have some more results, hopefully this time next year when Ariel invites me back, is to take our pictures of, from the kind of classic set of numerals here, handwritten, and project them, paint them, if you will, onto the cortex of the neural organoid. And so in this case, here's an 8. We can change the number of nodes which are stimulated by this particular 8, if you will, to make them systematically larger. Or we can train the model and then rotate that 8 to a different portion of the organoid and watch the chaos. See if it could still learn and how far we have to rotate it before it completely fails. So we think these are pretty cool. The fundamental notion of the application of that telegrapher's equation here is something called the reaction diffusion relationship, which is a partial differential equations which describe the evolution of chemical species over space and time. We're borrowing this idea to show how activation would spread across the organoid as well, and create these very interesting patterns that have been reported in fMRI studies of brain activity. That is of spiral waves, standing waves, soliton waves, and a number of other different traveling wave phenomena. So we're really quite excited about this, to be able to have this as a model system informed by some of our neuroimaging data analytics drawn from large-scale databases to be able to pursue these. So in summary, we're trying to use this idea to create neural networks which are more neuron-like, based upon foundational neuroscience, leveraging recent concepts of microstructural modeling, and as three-dimensional structures that we can teach to learn different things. And so again, this is all driven by voltages, not random weights. Moreover, I should say that the weights that get emerge from these systems are not just some random dimensionless weight. It's actually a conformational change in the underlying morphometry of the organoid. We can manipulate and change the amount of the G ratio that's required to transmit information at a particular speed. We can make those fibers fatter. We can make them skinnier. We can change all that stuff, or rather have the system learn how to do that so that you end up with, start with a sphere, you may get something completely differently shaped at the end, and we're undergoing that now. It's really kind of counting on a number of different things like dynamic systems, network theories. And anyway, we think they're pretty cool. And this was something, again, which was, we started with data. We worked through like what would happen in clinical cases and then how these might be something that we could then model. So just some very simple conclusions. Then if we have time left, we can do some questions. Really, to me, the brain is the source of all data. If you don't have a brain, we don't have brains collectively, there's no data, right? It's like if a tree falls in the forest. If there's no brain, do we have any data? And brain science itself is a data science and vice versa, but we shouldn't be surprised, right? Given how the brain has changed and evolved and what it's dependent upon in order to get to its current size from evolutionary speaking, counting and calculating is probably a major component of that. And in spite of things like rough beginnings to cultures of sharing, neuroimaging represents a relative success story of that. The origins of that are living in my basement in Virginia. But we need to do more than just doing statistical analyses and applying machine learning models. We really need to think principally, be principled about the models that we use and what we're trying to do in order to describe how brains are built and how they operate. And I'm hoping that you guys will do that. Ideas from digital twins and related methodologies may be very important for taking these ideas, the results from your analyses, and then turning them into something that you can manipulate and make inferences about. And this helps with your explainability. So it's something everyone should have some interest in. And in so doing all of these things, we can really use the brain and bring that brain into focus using a science of data. And it's really through events like this one, like this Neurohack Academy. Here we are in Seattle, Washington, my hometown, where this kind of thing can happen. And I really hope that you'll want to pursue that. I've written a ton of stuff on this kind of idea or areas peripheral to it. Please look that stuff up. If you want to talk to me at lunch or after the coffee break or whatnot, I'm happy to. These are my crew. We've been funded for a number of years by the NIH on various aspects of this stuff. Again, come visit us at the University of Virginia. We'd love to host you. And thank you very much.